Okay, so welcome back to this next video in which we are discussing cutaneous nociceptors. Okay, so we're currently in the process of looking at the structure of the skin. Okay, so we've seen that the skin has these four major layers. We've got the epidermis, which is made up of keratinocytes. We've got the dermis below, which is mainly made up of connective tissue, mainly collagen and elastin proteins. Underneath that, we've got the subcutaneous tissue, which is mainly made up of fat cells. And then underneath that, we've got the deep fascia, which is another layer of very dense connective tissue. Okay, now we were just discussing that you had connections between these two connective tissue layers. So between the dermis, which is made up of collagen and elastin, and the deep fascia below, and that these are called skin ligaments. Okay, now the formal old anatomy name for skin ligaments is to call them the ligament retinaculum cutis. Okay, so skin ligaments are known as ligament. So if you find a single skin ligament, so I'll take the S off, so a single skin ligament, it will be called ligament retinaculum, okay, and then cutis. So the ligament retinaculum cutis. That's the old anatomist's name for a skin ligament. Okay, and basically they're just connective tissue connections between the two layers of connective tissue. Okay, right. Now, what other structures do we have to discuss in the skin? Well, we can add on the sweat glands onto our picture, and we also need to discuss the whole purpose of this video, which is the cutaneous nociceptors themselves. Okay, so we'll just add a sweat gland. So basically, a sweat gland is just a little tube that opens up right on the surface of the skin like so, and usually the actual portion which produces the sweat is right down in the subcutaneous tissue. Okay, so you'll have a little coiled up tube here like so, which will actually produce the sweat. Okay, and this portion is known as the sweat gland here. So we've got our sweat gland down in the subcutaneous tissue, which is basically just a coiled up little tube, which then has this sweat duct, which goes and releases onto the surface of the skin, and this will just produce salty water. And obviously the sweat glands become active when you are presented with a thermodynamic challenge. So when the external temperature is too high and you're at risk of body temperature going above what we want it to be, then you will sweat to put water on the surface of the skin, and the water on the surface of the skin can then evaporate, and when water on the surface of the skin evaporates, it will take with it a lot of energy, and that will remove heat energy from the skin is the idea, okay, and therefore cool you down. Okay, right, so now let's show on this picture the cutaneous nerves. Okay, so let's put in a cutaneous nerve, and specifically we're interested in cutaneous nerves which are going to detect painful stimuli and fire action potentials in response to painful stimuli. Okay, so basically it will come in like so, and then it will branch out many times, like so. So it will have loads of little branches, and some of these branches will go right up into the epidermis, some of them will remain in the dermis like so. So basically these cutaneous nociceptors, not all of them will be nociceptors. I mean, there are cutaneous nerves that aren't nociceptors, but we're specifically interested in cutaneous nociceptors. So we'll suppose that this is a nociceptor. It's a neuron which is specifically going to detect noxious stimuli and fire in response to those noxious stimuli. So let's say this is a cutaneous nociceptor. Okay, right. So let's now talk about the examples of noxious stimuli which cutaneous nociceptors will react to, and then we'll talk about the different types of primary somatosensory neurons and which types can actually be nociceptors. Okay, right, so let's start off with examples of noxious stimuli, and these are pretty common sense. Okay, so what hurts? Okay, well basically extreme temperatures will activate nociceptors. Okay, so the extreme temperatures will be our first example of noxious stimuli. And basically, this can either be too hot or too cold, basically. So, what sort of temperatures start to activate our nociceptors? Well, basically, hot is very, very easy. Generally, it's if you go above 43 degrees Celsius, then you will start to activate your cutaneous nociceptors. Okay, cold is much more difficult. There's much more variation in the human population. Okay, so hot is pretty much set. Everyone's nociceptors pretty much become active at 43 degrees Celsius. Whereas if you do an experiment where basically you put a little sort of metal block on someone's arm, okay? So let's say this is someone's arm here, and basically you put a little special metal block called a thermode onto their arm, and basically it's just a metal block which has been either heated up or cooled in a water bath to a certain temperature, basically. And you put it onto their arm and you ask them to say whether it's painful or not. Okay, so if you do this experiment where the thermode is hotter than 37 degrees Celsius, okay, you find that generally people report that the thermode becomes painful when you finally go past 43 degrees Celsius. So you'll be continuously varying the temperature of this thermode. So you might start off at 37 degrees Celsius, raise it to 38, 39, 40. Basically, you'll find that people will start reporting that they find it painful when it goes above 43 degrees Celsius. If, on the other hand, you do it for going too cold, then it's really variable when people start to actually report that it's painful. But generally, people start to report that it's painful if it goes below 15 degrees Celsius. Okay, so going too hot, there seems to be a set point where people start to find it painful, whereas going too cold, it's hugely variable, basically, much more variable than going too hot. Okay, right, so extreme temperatures are examples of noxious stimuli, okay, which will activate nociceptors within the skin. In addition, intense pressure, okay, so if you press down on the skin too hard, then eventually it will cause pain, basically. Okay, and this is because the cutaneous nociceptors are becoming activated. In addition, tissue damage will cause pain. So if someone punches you, that will cause a lot of tissue damage, and that will cause a lot of pain. And you might say, well, maybe it's the intense pressure, but you know, the intense pressure will be relieved after a while. You know, the intense pressure will only be there for a very short time. After that, you know, you've no longer got the fist against your skin. So what continues to cause the pain? Well, it's because it's caused tissue damage. So how does tissue damage activate the nociceptors? Well, basically, what's going to happen is that when your tissue becomes damaged, it will release all sorts of chemical mediators. Okay, so damaged tissue will release all sorts of chemical mediators. What examples of chemical mediators can I come up with? Okay, so basically, if you imagine breaking open a cell, okay, so let's say we've got a cell here, and we break it open, then basically certain things are going to be released from the cytoplasm of the cell. One of the key things is ATP, adenosine triphosphate. Okay, so usually ATP levels within the extracellular fluid are kept extremely low. Okay, so usually ATP is extremely low. So if ATP suddenly goes up in the extracellular fluid, you know that that's because cells are dying. Okay, so ATP is an example of a chemical mediator. In addition, you might recall that in the lysosomes of cells, okay, so here's our cell nucleus, and this is supposed to represent a lysosome. In the lysosomes of cells, you have enzymes which break down proteins known as proteases. Okay, when you break the cell open, these proteases are going to spill out. Okay, and basically these proteases can activate all sorts of signaling cascades within the extracellular fluid, and they can result in the production of chemical mediators which then will activate nociceptors, basically. In addition, finally, we know that cells contain a large amount of potassium ions. Okay, so when you suddenly burst open a cell, that will release a huge amount of potassium into the extracellular fluid. So remember that the normal extracellular concentration of potassium is 4 millimolar, whereas the intracellular concentration of potassium is usually around 155 millimolar. So usually potassium concentration is much higher inside the cell. If we suddenly break the cell open, all of this potassium is being released into the extracellular fluid, and it will raise the concentration of potassium in the local extracellular fluid. This can also activate nociceptors to fire. Okay, so these are examples of some of the noxious stimuli which will be produced by damaged tissue, basically. Okay, so noxious stimuli we will now define as things that are capable of activating nociceptors. Okay, and what we'll discuss, not in this video but much later on in this playlist on pain, is that activating nociceptors isn't necessarily actually going to cause pain. It's far more complicated than that. You can have a painful stimulus present within some cutaneous tissue. It can be activating nociceptors, but that might not actually be getting all the way up to the brain to cause the conscious experience of pain. Pain is one of the it's one of the sensory systems which has the greatest capacity to be modulated, basically, by higher systems. Okay? When we compare it to other forms of somatosensation, such as thermosensation and mechanosensation, those we can't really cut off our ability to sense mechanical stimuli. Okay? But you can modulate your pain experience, basically. You can modulate the effect that nociceptive stimuli are going to have on the brain. Okay, right, so how nociception is actually transferred into a painful stimulus is much more complicated, and it might be that you can have nociception without actually getting the painful stimulus. And in addition, you can also have you can also experience pain without having the nociceptive stimulus. Okay, right, so what I now want to turn our attention to is the different classification of neurons, basically, and which types of neuron can actually be nociceptors. So, basically, we have a huge number of primary somatosensory neurons. Okay, so a primary somatosensory neuron is just a neuron which transduces the sensory stimulus into an action potential and then takes that stimulus to the central nervous system, and it usually takes it all the way into the spinal cord. Okay, so this is a primary somatosensory neuron. Okay, and basically you can classify primary somatosensory neurons into 4 separate categories. Okay, and these categories are based on anatomical features, basically. They're based on how thick these primary somatosensory neurons are. So basically you just look at all the different primary somatosensory neurons which are going into the spinal cord, and you find that they range hugely in their diameters, basically, and you then can categorize primary somatosensory neurons into the categories depending on their physical characteristics. Okay, so the 4 categories that primary somatosensory neurons are usually categorized into are A alpha, A beta, A delta, and then also C fibers. Okay, and I want to stress once again that this categorization is done on an anatomical basis, not on a functional basis. Okay, so A alpha fibers are massive great fibers. Okay, so let's try and draw these here. So let's say this is the actual neuron here, And basically, let's say this is the axon, okay? So remember, if I just draw you a little picture of a neuron. So the neuron looks like so. Okay, here's the cell body. Well, actually, the sensory neurons aren't going to look like this. The cell body is not going to be like this. This is a typical motor neuron. However, basically, the bit that we are looking at is the axon, and I'm taking a cross-section through the axon. Okay, so basically, the axon diameter of A alpha neurons is around 20 micrometers. Okay, and also the axons themselves will be surrounded by myelin sheath. Okay, so they'll have Schwann cells surrounding them, and not only that, but the Schwann cells will actually produce myelin, and therefore they become myelinated. They get a myelin sheath surrounding them. Okay, so remember, the myelin is a separate entity that the Schwann cell has to produce. It's not a case of the Schwann cell just wraps around you and you then have a myelin sheath. No, the Schwann cell has to produce the myelin. Okay, so these A alpha fibers, they are lucky enough, their Schwann cells actually do produce the myelin. Okay, so this is called a myelin sheath here. Right. The A beta fibers are then smaller than the A alpha fibers. They're about half the size, so let me try and show their diameter as half the size. Okay, so here we go. And they are around 10 micrometers across. Okay, and these are very rough estimates, basically. Okay, of course, if you've got something that's around, let's say 16 micrometers, this will probably be categorized as A alpha. Okay, whereas something around 13 micrometers, this will probably be categorized as A beta. Now, the A beta fibers are myelinated as well. Okay, so they're just slightly smaller in diameter than the A alpha fibers. A delta then halves again. Okay, so here now is our A delta. It's 5 micrometers in diameter. Okay, so its axon is 5 micrometers in diameter. And you might be wondering, well, what's the significance of all of this? Basically, larger diameter axons conduct action potentials faster than small diameter axons. Okay. In addition, having the myelin sheath will speed up the speed of conduction, basically. Okay, so all of these will conduct reasonably fast because they've got the myelin sheath around them, and the A alpha of them all will conduct the fastest because it's got the biggest diameter, followed by the A beta, followed by the A delta. C fibers are absolutely tiny, okay? They are 1 micrometer in diameter, okay? And they do not have a myelin sheath. Generally, the C fibers will be found in groups together, so you'll have loads of C fibers all together like so, in what's known as a fascicle, and they'll have 1 Schwann cell surrounding them all, basically. So here is the 1 Schwann cell that surrounds all of the axons of all of these C fibers. Okay, so they do have a Schwann cell surrounding them and looking after them, but the Schwann cell doesn't produce a myelin covering around them, and they all have to share a Schwann cell, basically. So this structure of many of them all clustered together is known as a fascicle. Okay, right. So that's the 4 different categories of primary somatosensory neurons: A alpha, A beta, A delta, and C. And I want to stress once again that this is an anatomical categorization. All they have done is they've gone and looked histologically at the neurons that you find if you take a cross-section of a nerve, and they've categorized them into these 4 groups based on what they look like when you take a cross-section of their axon. Okay, there is no functional divisions here. Okay, it's just merely anatomical divisions. Okay, right. So, what we now want to ask is, nociceptors, can they be any one of these types of neuron? Okay, and the answer is almost. They can't actually be A alpha. You don't generally find nociceptors which would fall into the A alpha category, but you do find nociceptors which would fall into all of these 3 categories. And I should just say the C fibers, which are unmyelinated, they conduct the slowest of all the primary somatosensory neurons. Okay, that's because firstly their diameter is the smallest, and secondly they don't have the myelination. However, their conduction isn't that slow. They generally have a conduction speed of around 0.5 meter per second, which isn't slow when you think about it. That's quite a fast conduction speed. You know, that will generally be the length of your arm in a second. Okay, so it's not bad. Okay, so A beta, A delta, and C fibers can all be nociceptors, or rather, we find nociceptors which would be categorized into all of these three categories. So you can find nociceptors which would fall into the A beta category, you can find nociceptors which would fall into the A delta category, and you can find nociceptors which would fall into the C fiber category. Okay, right. So, we'll call it there for this video, and we'll continue our discussion in the next video.